Hi everyone, welcome back to my video presentations on translation. In this series, we'll be discussing the mechanism of translation in eukaryotic system. In the previous videos, we have seen the mechanism of prokaryotic translation in detail. Hence, in this part, we'll not be elaborating much on already discussed part. Since both the mechanisms are almost similar in various stages. So we'll be focusing on the mechanisms which are a little different from that in prokaryotes. See, we know that there are certain factors and certain molecules which are different in eukaryotes, and also the location of translation is different in eukaryote, which occurs in the cytoplasm. Unlike the location is the same in the prokaryotes and eukaryote. That means after the processing of the mRNA will be transported into the cytoplasm. But in prokaryote there is no processing. It occurs together with the transcription. But in eukaryotes, the processed mRNA will be transported to the nucleus, into the cytoplasm from the nucleus, and there the translation occurs. So we'll discuss the translation mechanisms of eukaryotes in detail in the following presentation. When we look at the mechanism wise, the same stages are there in eukaryote also. There is activation, initiation, elongation and termination. So we will not be focusing already discussed part of the mechanism. So the activation part is having the similar reaction to that in prokaryote. Right, that means there is an amino acid which is charged, which is activated by addition of an AMP to form amino acyl AMP, then followed by charging this amino acyl or acylated amino acid into the tRNA molecule to form amino acyl tRNA. The only difference is that the tRNA involved in the beginning or in the initiation is not the tRNA F-met but tRNA met, and we use a subscript I to denote the initiation. So the tRNA is tRNA met I, and the amino acid is also not formyl methionine but normal methionine. Okay, so that is the main difference in the beginning stage. Otherwise the mechanism is the same to that in bacterial systems. But when it comes to the initiation, the mechanism is slightly different. The initiation has been subdivided into 4 stages. First one is a ternary complex formation, and this is actually the complex formation of the tRNA and the initiation factors. Then second stage, these ternary complex will be loaded onto the 40S ribosomal subunit, that means the smallest subunit of the ribosome, and then followed by scanning and AUG recognition. Then finally the larger subunit will attach to form the initiation complex. So this mechanism is slightly different from that in prokaryote. Even though the concept is the same, the main difference comes in the third part, that means the scanning of the AUG sequence. Because in prokaryotes, the translation usually occurs coupled to the transcription, but in eukaryotes it is not like that in prokaryotes. The free mRNA is there in the cytoplasm, and which usually forms a structure or which usually forms a circular structure due to the binding of the various factors involved in initiation. And then what actually do is, this ribosome can scan through the mRNA to locate the initiation codon, that means the AUG. So once AUG is located, then the initiation occurs. Okay, and moreover since it is having a circular pattern, the same mRNA could be used for multiple production of protein by the same ribosome. Okay. So this is also a difference in prokaryotes and eukaryotes, that in prokaryotes normally multiple ribosomes will attach on the same ribosomes, same mRNA, and you can see a polyribosome structure. But in eukaryotes it's usually there is no polyribosome structure, but instead the same ribosome will do the translation of an mRNA multiple times. So there you can see certain differences. And this is the list of initiation factors involved in eukaryotic translation initiation. You can see there EIF1, 1A, 2, 2B, 3, 4A, 4B, 4E, 4G, 4F, 5 and 5B. So a huge list of proteins are involved in the initiation, unlike in prokaryote. This is the first step, that means the ternary complex formation. Here the EIF2, which is homologous to the IF2 in prokaryote, bound with the GTP, will form a complex with the first tRNA, okay, which is having the methionine. So you can see there, this is the ternary complex, which consists of the tRNA with methionine, then EIF2 and GTP. This complex is called the ternary complex. And in the second stage, this ternary complex is loaded onto the small subunit of the ribosome, that means into the 40S subunit, and there forms a 43S intermediate complex. This is otherwise called a pre-initiation complex. Here we can see the major initiation factors with their corresponding function. EIF3 binds to the 40S subunit and block the premature binding of 60S subunit, just like IF3 do in prokaryotes. Then EIF1 and 1A are responsible for specific binding and prevent binding of tRNAs and other factors to the A site of the ribosome, which protect the A site during initiation phase. Then the 4F multi-subunit complex, which contains 4E, A and G. Then 4E is the cap binding protein, which binds to the 5' cap. Then 4A is a helicase enzyme, which unwinds any secondary structures that might have formed in the RNA molecule. Then 4G binds to the 5' tail through the poly-A binding protein of the mRNA. And this, so you can see that the 4F complex attaches both the ends of the mRNA to form a circle. That means 4E binds at 5' cap, 4G binds at 3' end, right? And the 4A together with 4B usually unwinds any secondary structures formed. So you will get a circular structures. Okay. So binding of both the ends to the protein creates this circle, and there is a kind of proof check also being done through this, because both the ends of the mRNA are essential structures for a successful translation, which means if an RNA molecule has lost any part, maybe a 5' part, then that mRNA will not be translated. That means any truncated mRNA will not be translated by the system. So this is a proof checking. So this is the mechanism. You can see there is the 4F complex coming in one side, and the other side you can see the ternary complex. The ternary complex with the 43 will form the 43S complex, pre-initiation complex. Then the mRNA will be positioned into the pre-initiation complex with the help of the 4F. Okay, so this is loaded now. So these are the first two steps. One is the ternary complex formation, and second is the loading of this mRNA to the 43S complex. This is another image showing a little more simpler image. You can see there the poly-A tail and the 5' cap are joined to the same complex. and this is the 40s subunit at the top. Okay. And when we talk about the initiation, in eukaryotes we can see two different strategies for initiation. One is called cap dependent initiation. That is what we will be focusing on, which is depending on binding the cap protein and the poly A with the same 4F complex and circularization. Then in the other one, cap independent model, there is no such cap proteins associated, but instead the ribosomes directly enter into the mRNA's codons, right? So that's what called the cap independent model. Then the next stage of initiation is the scanning for AUG sequence within the mRNA molecule. The 43S complex usually scans along the mRNA from the 5' direction to the 3' and will search for AUG start codon. So this movement continues until the AUG is encountered. So once the AUG is found, that will halt there. And usually the first AUG is usually found embedded in a sequence called Cossack sequence. You can see there an AUG sequence within the ACC AUGG sequence. So this Cossack sequence will help in recognition of the AUG by the 43S complex. And the specificity of the reaction will be controlled by the EIF1 and 1A. That means which blocks the entry of any other tRNA molecules into the A site and provide some specificity to the screening or the scanning mechanism. Now you can see the codon-anticodon interaction. These are the same seen in prokaryotes. The tRNA with its anticodon can bind onto the codons in the mRNA at the P site. And there will be GTP hydrolysis followed by the release of phosphate. And now this complex becomes a 48S initiation complex, right? So this complex now is ready for the next round, that means for accepting the 60S subunit of the ribosome. And that is the last stage of initiation. The 60S subunit will be recruited along with a factor EIF5 GTP complex. So this EIF5 GTP complex causes release of other initiation factors from this complex, and there will be a release of GDPPI, EIF1, EIF1A, then EIF5, everything will be released, and you will get only the 40S, 60S, the tRNA and mRNA as a complex, right? And of course the poly A tail and the poly A tail binding proteins may influence the joining of the 60S subunit to the structure. This will give you an idea about the ribosome assembly. So once the EIF1A and EIF1 are released from the ribosome subunits, the A site is now ready to accept the next amino acyl tRNA, and that will mark the elongation process. There is an internal recycling process also. This is also similar in prokaryotes. There is the IF2 was recycling. Here also you can see the EIF2 will be recycled with the help of EIF2B. That means EIF2 GTP will be reformed, and this is because this is required for further cycles. Then the EIF5B also undergo the similar process of recycling, and this shows the overall mechanism of translation initiation in eukaryotes. You can see the entire events. In the beginning you can see the tRNA combined with amino acid, then ternary complex formation followed by the 43S mRNA complex formation by attaching to the 40S subunit. Then there is 48S complex formation, then that also followed by the attachment of the large subunit to form the final initiation complex. So now this system is ready for the next round of translation, next stage of translation called elongation.